Hello.
Hi everyone, how are you today?
I'm very well, thank you. How are you, Katerina?
Can you check if the link works for you guys? Thank you.
401 unauthorized.
Okay, I'll have to tell. Yeah, I asked. Okay.
Is there a way to
We'll.
adjust the permissions maybe on your side to make it public?
It's not mine.
I see. Okay.
Yeah, but I'll add the paper for now, and then we'll have to do it live. Let's see.
Hi, Dennis. How's things?
Hi, Jamie. Good to see you. Pleased to be here. How are you doing today?
I'm quite well, thank you, actually. Was reading through some of the paper, trying to understand what I could, and educating myself on biology a lot. But it's fun. Very fun.
Any highlights you want to share?
Um, you mean about biology in general or the paper specifically?
Well, you said you were learning a lot. Do you want to share something you
Oh,
learned?
oh, right. Okay, well, I didn't actually know that we have around 200 cells, or 200 types of cells in our bodies. I didn't know that one.
Very cool. Did
Yeah.
well, what else?
Um, I didn't actually, was not totally sure what a zygote was until I had to look it up.
Hi Alejandro.
Oh,
How are you? Could you please change the settings of the Google Drive link? I just sent you an email, but because we cannot access it, it needs to be, in order for it to work, that everyone can view it with the link. If you click on share, and then thank you so much. We wait a couple of minutes to give people time to arrive, and then we can start. So no stress.
Sure, just will take 1 minute. Thank you.
hello, Doctor. My name's Jamie. Thank you so much for coming. Am I coming through okay?
Dr. Shah, welcome. Good to see you.
Pardon?
Hi Dennis, hi everyone.
Hello. How are you today? Welcome.
Welcome Alejandro.
Thank you.
Alejandro, just so I'm getting it right, is your surname, is it Castejon or
Yeah,
Castejon?
it's Alejandro Aguilera Castrejon.
Castrejon. Okay, Castrejon.
Yes.
Good. I wanted to make sure I got the pronunciation right because, you know, I'm trying to learn Spanish myself, so I'm practicing with the sound.
Ah,
It's actually much easier for myself being Scottish because we say loch. That comes in very handy. Looking at some of your paper as well is very, very interesting. Looking forward to this talk.
Okay,
thank you.
let me switch then to the link, and then we can start. Oh, you. Okay. Oh, you sent me the present. Okay. One more minute. In the meantime, I can introduce you, and then usually Jamie asks a few questions. Like a few general questions, and then we start the presentation. So in the meantime, I'll download the presentation.
Sure.
Right. Yeah, thank you everyone for coming to our Science Society meeting today. I'm very honored to have here our guest speaker with this amazing research. So let me tell you a little bit about him. He did his master's in life sciences at the Weizmann Institute, and his bachelor thesis he did at the Instituto de Fisiologia Cellular in 2016, and he also did his PhD in life sciences at the Weizmann Institute of Science, and he is in the Jacobs Hannas lab for pluripotent stem cell research and epigenetic reprogramming. And he recently published this really exciting study, and we are very happy to having you here. And please, Jamie or Denise, go ahead with more general question. Thank you.
Thank you very much. Can I just ask, would you prefer Alejandro or Castejon? Dr. Castejon? Do you have a preference?
Alejandro is fine. Yeah.
Thank you very much, Alejandro. I really like this part because we all get to know a little bit more about you. I was curious, has biology always been your main passion? Like, or did you sort of get here from some kind of circuitous route?
So actually, I always liked biology. I was actually first interested in kind of working with animals, in the jungle of Mexico. But then I discovered that I like developmental biology, and I switched to this field. But I was always interested in biology.
And what parts of the jungle did you like the most when you were there?
In the south of Mexico, there is a very big jungle, and I still like to go hiking or something, but I discovered that I don't really like to work with the animals. I prefer to work with the embryos.
Ah, okay. So you just want to know what they're made of, yeah?
Yeah.
And was this curiosity, was it what drove you to this particular branch of study here?
Mm, so I was in Mexico. I was interested in regenerative medicine. So when I was looking for a lab to do the master, um, I was actually trying to work with the cellular reprogramming, to take somatic cells and reprogram into pluripotent. And my advisor here is actually very famous for that. So I actually came here thinking about working on that, but then he offered me to work in this project of trying to grow the embryos ex utero, and I discovered that I like it a lot, and then decided also to stay for PhD.
That's interesting. So he was a big influence on what brought you to where you are now?
Yes, for sure.
He must have seen that your talents were quite well suited to this particular thing. Then, were you first reluctant to take this on, or from the very get go were you thinking, "Wow, actually, this sounds incredibly interesting"?
So first I was working with embryonic stem cells for like 4 months, but then I actually switched. He offered me to work in this project, and then I decided to switch. Immediately I thought it was very interesting, and I really like it a lot. Still now after 5 years, I still like it.
Did
Great.
you
Then,
feel you? Oh,
oh,
so
yeah.
I was going to ask just one more. I was just going to
Yep.
say, do you still feel you're learning the whole time when you're doing and when you're working in this particular field? Because I can see you're achieving a lot in it, but do you still feel you're all the time learning?
Yeah, I think every time, because it's still like we advance a lot, but let's say it's not finished, right? Like
Hmm,
we still have a long way to go if we would like at some point to see a living mouse coming without
that'd
alive
be
without a
amazing.
mother. So it's all the time, and you learn also about kind of what the embryos need on metabolics, or also the signaling or the growth of the embryo. So it's a lot of, or even physics, mechanics of how the cells interact. So there's always something new to learn.
Fascinating. Thank you very much. Okay, take it away, Katarina.
Yeah, I think the PowerPoint is working now, so yeah, we can start. Thank you.
Okay, great. I will start. Thank you for the nice introduction. Just wait 1 second. I need to open my presentation. Yeah, that's it. So, my name is Alejandro, as you know, and I'm doing now my PhD in the lab of Jacob Hanna, the Weizmann Institute of Science. And I'm really happy to have the chance to talk to you today about my project, trying to create a system to grow mammalian embryos outside of the uterus. So I work with a mouse, and just in general, the mouse development lasts for 19 days, in contrast to the human, which is 9 months. In mouse, is very short. And if you look at the slide number 2, so you will see a kind of a scheme of the mouse embryo, how they look at the different days. And in general, mouse development is divided into pre-implantation and post-implantation development. And the implantation in the mouse happens in the day 4, with in this day, the mouse embryo that looks like a sphere, that is called the blastocyst, implants into the maternal uterus, and then it undergoes a change in morphology, and it generates something that is called the egg-cylinder embryo in day 5. And this cylinder embryo has only basically 3 cell types, but these cell types are still not differentiated. So only in the day 6, when the process of gastrulation will start, in which basically the embryo will go from cells to an embryo, some like the establishment of the different axes, where the head will be, what will be the tail, what will be the left, right, what will be the ventral part, the dorsal. So this starts the process of gastrulation. And in the case of the mouse, the day 6 is considered early gastrulation, and the day 7 late gastrulation. So it lasts from day 6 to day 8. And from the day 8 onwards, the process of formation of the organs will start. And by the day 13, most of the organs will be already established in the embryo, and we will and a period called the fetal growth will start, in which basically all the organs only need to mature. And you can see in the day 13.5, you can already see very well the morphology of the mouse, and it will be born at 19 or maximum 20 days. And as you can see, the process of post-implantation actually lasts for 15 days, which means it comprises 75% of the total development of the mouse embryo. And still we know a lot more about the pre-implantation period than about the post-implantation. And this is because in the post-implantation, everything happens inside the maternal uterus. So, and this, of course, makes it inaccessible to observe. We cannot see how the embryo grows inside the maternal womb. Also, we cannot do experiments, and it's also not possible to take one embryo and then do some experiment and then put it back into the mother. This is impossible to do. So a solution for this will be to create a system to grow mouse embryos in a petri dish or outside of the uterus. And here, if you go to the next slide, slide number 3, you will see images of the real mouse embryos, how they look at different days, from day 0 to day 11. And in the case of the culture of these embryos, actually the culture of pre-implantation embryos from day 0 to 3 is already very well established, as well as the culture of what is called in vitro implantation. So you can see from day 0 to day 4 is the pre-implantation, and we can also model in vitro implantation from day 4 to day 6, and this is already established for mouse, for monkey, and even for human embryos. However, when we go to post-implantation stages, it has been much more challenging to establish such cultures. And actually, the most commonly used method is called in rotating boros culture, and it was established almost 50 years ago by Dennis New at Cambridge University. And however, these methods, first of all, were not changed in 50 years. They are also short term, since maximum you can keep the embryos for 36 hours alive. They are also inefficient, since many of the embryos that you take will be abnormal. And because of these reasons, they were not widely adopted by the community, by the developmental biologists. But still, these methods actually are very important because they allowed us to basically do research and identify many milestones in mammalian development. For example, people identified in 1994 some organizer of the mouse embryo called the node, and also people were able to create fate maps of the mouse embryo using these short-term cultures. But we wanted to expand it, so if you go to the next slide, slide number 4, you will see, first of all, again, the scheme of the mouse embryo from day 5 to day 11. And then we wanted to expand this culture, and the first thing that we did is to try to expand it starting from the day 7 of the embryo until day 11. And for that, basically, we use the previously established royal culture incubator, and we adapted to this incubator gas regulation model that basically what it can do is allows you to control very precisely the level of oxygen and also the level of carbon dioxide, but not only that, also the pressure of the gas, which is actually we found that is one of the most critical parameters for the proper growth of the embryos. And this, in combination with the uh, uh, changing the levels of glucose through the culture, and also again modulating the oxygen, and the use of a specific ex utero culture media that has a high percentage of rat serum and human serum, allowed us to grow the embryo from the late gastrulation stage from day 7 until day 11, so for 4 days. And what is important about this culture is also that it's very efficient. About 75% of the embryos that you take in the day 7 will reach the day 11. And this also works across several lines of mice. So it's a very stable system. And if you go to the next slide, you will see that we perform a detailed morphological analysis of these embryos. And on the top left, you can see the in utero embryos and all the different structures that they have. And on the bottom left, you see the ex utero embryos, and they are really equivalent to the in utero. And on the right, you will see some immunodetection of different proteins that are expressed in the different tissues of the embryo. We analyzed 11 different proteins, and in this case, I'm showing you only one. It's a protein that is mainly expressed in the neural tissues of the embryo, and you can see that the distribution of this protein is basically identical in the embryos that are grown in utero and also ex utero. So if you go to the next slide, you will see that, so we already established the system to grow from day 7 to day 11, and then we wanted to expand it one day more, but it's backwards. So basically to start with the early embryo on day 6.5. And then for that, we also tried to use the roller culture. However, no matter what we tried, it didn't work for us. So then we decided to use a static culture. Basically, just put the embryo in a plate and leave it there in the incubator. It's not rotating all the time as in the roller culture. And we used some conditions that were previously established. However, we found that these conditions have a low efficiency. So we decided to also optimize some new conditions to grow the mouse embryo first from the day 6.5 to day 8.5 using a static plate. And we managed to find these conditions that are actually extremely robust, that almost all the embryos that you take in the day 6.5 will grow properly to the day 8.5. And then if you go to the next slide, I'm showing you some live imaging of the mouse embryo. You can see, I hope you can see the videos. These are some mouse embryos that express a protein that is in red colors called TD tomato. And you can see basically that these embryos in a static culture, we can image them for up to 58 hours. And basically we are able to see the transition of the egg cylinder, basically from a pluripotent or as egg cylinder into an embryo that already has a head and a tail. And if you go to the next slide, then you will see that we also, of course, perform an analysis of the different developmental markers to see that the embryos are actually developing properly also at the molecular level. However, this analysis with antibodies is limited always to very few markers. We analyze, as I said, on 11. So it could be that there are some missing cell types that we didn't manage to find. So because of that, we decided to perform an analysis of the whole transcriptome at the single cell level in the embryos at this stage. Just to not have any bias and analyze the whole or all the genes that in the embryo. So if you see the distribution of different transcriptional states, basically every dot that you see in the graph is a cell, and you see that the distribution of the ex utero in plus day 2, which is equivalent to 8.5 in utero, is very similar, meaning that the cells that you find in utero, you also find them ex utero, and in the profile of cell types, it corresponds to the what we expect to see in the embryo in the day 8. So with this we can show that actually these embryos actually are able to make all the cell types that are found in utero. We also find them ex utero. If you go to the next slide, then we already established the protocol to grow from 7 to 11 in the roller culture, and also from 6 to 8 in the static plates. And just by transferring the embryos that come from the static plates from 6 to 8 to the roller culture, we're able to extend this culture for up to 5 days. So from the early gastrulation is 6.5 for up to 5 days, with efficiency close to a bit higher than 50%. And you can see there in the images the morphology of the embryos at different days. And importantly, we can also take the embryos even 1 day earlier, so in the day 5.5, which is basically the first day of post-implantation development. And the embryos can develop for up to 6 days outside of the uterus. And you can see how they basically are transformed from a cylinder that has only 3 cell types into an embryo with many, many tissues. And the efficiency is close to 20%. So now if you go to the next slide, you will see that also in these embryos we perform a single cell analysis, and you can find that the overlap between the green dots of the ex utero embryo at day 4 and also in utero day 10.5 is basically equal. And the profile of cell types that you find in these embryos already correlates with the advanced organogenesis. So we see already many different cell types of kind of more mature organs. And because we did this single cell analysis, if you go to the next slide, we can also analyze very precisely if for example there could be that some organ actually has more cells in utero or less cells compared to ex utero. So we wanted to see maybe they have, for example, say a bigger heart or less cells in the brain. So we wanted to see it with the single cell analysis, and we actually found that if we analyze the proportion of different cells, of the number of cells in the embryos, we see only a small difference in 3 of the 20 different cell clusters that we found. And also, if we compare if they are genes that are differentially expressed ex utero compared to in utero, we actually found that there are very, very few genes that are expressed differentially, and only in 2 clusters. You can see them in red. The actually one of them is the erythrocytes, and the other one is the cells of the heart, the myocytes. And these actually have only 2 and 8 up to 2,000 genes that we analyze that are variable. And we think that the fact that the cell types that have this variability on the gene expression are the cells of the heart and some cells of the blood, the erythrocytes. And so we think that this is consistent with the fact that actually the embryos already are suffering because of insufficient oxygenation and also deficiency in the nutrient supply. Because actually at this day, and the day 10.5 or 11, is when the embryos will become more dependent on the placenta of the mother. So in this case, they don't have placenta. So mainly the heart and the blood are organs that kind of start to suffer first, and we see the changes in the gene expression. So if you go to the next slide, you can see that basically in this culture we can take an embryo from the pluripotent epiblast, basically one cell type that will give rise to the whole body of the embryo, and it will grow outside of the uterus into a fully formed embryo with a head, eyes, limbs, and tail. Uh, and you can see here what is the kind of the real change in size of the embryo. You both for day 6, and actually the change is more dramatic for day 5. After 6 days of culture, you can see that the embryo really changes in morphology and size. And all of this happens without any interaction with the mother. So the embryo is actually able to do it by itself. It's something that is called self-organization. And if you go to the next slide, then you will see that it's not enough for us to demonstrate that we can grow the embryos properly. We also want to be able to do experiments on them. So we wanted to prove that we can do modifications and the embryos will still grow properly and actually are able to analyze what is the result of those modifications. So this is a proof of principle. First, we perform electroporation of a GFP reported plasmid at day 8.5. So what we did is we took the embryo at day 7, and then the day after we injected the plasmid in the brain and incorporated the embryo. So uh, this helped for the plasmid to enter into the cells, and then you can see how the green signal of the GFP is expressed after 1, 2, or 3 days in a specific region of the brain. We wanted to do this modification, but also if you look in the right side of the slide, we also wanted to perform a gene editing in a more widespread manner. So for that, we injected lentiviruses with the GFP plasmid in the E6.5 mouse embryo, and you can see in the immunostainings of GFP that basically we can see the integration of this vector into most of the body of the embryo, and they are able to survive properly. So this system will actually allow us to later try to do some genetic modifications in the mouse post-implantation embryo. And if you look at the bottom of the slide, we also perform in total live imaging of the neural tube closure from day 9.5 and day 9 in the mouse embryo. So what we did here is we took the embryos at day 7.5. We put them in the roller culture and we grew them until day 9. And then at day 9 we opened the embryo and took it to the microscope. And you can see that the process of the closure of the neural tube in a mouse transgenic embryo that expresses the red protein T tomato. So we can do this imaging for up to nine hours, and we can see very nicely how the neural tube is closing. So now, if you go to the next slide, we also wanted to do something that is called chimeras. So basically, you can inject cells and see how they integrate into an embryo. And actually, this generation of chimeras is the golden standard functional assessment of the pluripotency of cells that we have in culture. And this is possible because we can inject cells into the pre-implantation embryo and then put this pre-implantation embryo back into the mother, and it will grow. It will grow, and we can analyze it. How the cells, if the cells were able to integrate into the embryo. And this is, if you look in the
Um, I cannot hear you anymore, Alejandro. I think you muted by accident.
sorry,
No
sorry,
worries.
sorry. Sorry. So I will come back. So as I was saying, we also decided to do these chimeric embryos by injecting pluripotent cells into the embryo, and this is done basically in, as you can see in the left, we can inject naive embryonic stem cells that resemble the pre-implantation embryo, and then put them back put the embryos back into the mother, and they will grow, and then we can analyze how the cells integrated into the embryo. However, this was not possible for the post-implantation embryos because we cannot put back into the mother a post-implanted embryo. So the only way to do it is with a culture that allows you to grow the embryo ex utero. So actually now we have a system in which we can analyze the pluripotency of the post-implantation embryo. And this is basically what we did. We took cells cultured in vitro, and we took also cells directly from other embryos and injected them in different positions in the embryos and performed this ex utero culture. And after we analyzed if these cells were able to integrate into the body of the embryo, and we can see that if you look at the immunostainings on the left side, uh, that after the one, if we inject these cells that express GFP, as you can see in green, after 1 day, there is a nice integration of the cells into the body of the embryo. However, at day 2, we already see very few cells, and you can see in the graphic that after 3 or 4 days, we see very few of these cells integrated into the body of the embryo. And this actually is it contrasted very well with the cells that we took directly from the E7.5 epiblast. After 1 day, you can see that in this case, the cells in red uh, of the 3D tomato are very well integrated into the body of the embryo. And even after 4 days, we found even embryos with up to 10,000 cells that are integrated into the body. So basically, this just shows that the contribution of the cells that we took from in vitro is lower compared to the cells that we took directly from embryos. So maybe there is a need to find alternative conditions to grow the pluripotent cells in vitro in order to make them to be integrated well in the body of the embryos. So basically, this is one conclusion that we got from this, and this in the cases made with mouse cells into a mouse embryo. But if you go to the next slide, we can do it also injecting human cells into the mouse embryo. And this is because just trying to analyze what it will be the behavior of the human cells inside an embryo that is alive. So to do that, we took human embryonic stem cells and differentiated them into microglia progenitors. You know, these basically the immune cells of the nervous system. And we injected them into the embryo at 7.5, and then we analyzed them at between day 9 to day 11. And you can see on the right, at the top right, the image that how this cells of course express GFP and how the embryos look in the day 0 after the injection. And if you look at the monostaining below, you can see that after 3 or 4 days, we saw a nice integration of these GFP green cells, and also they proliferated and then migrated to the brain. We found them mainly in the brain of these mouse embryos, and we perform, of course, the staining for GFP to find the cells, but also for a protein called NUMA that is actually specifically found in human cells, uh, just to confirm that these cells actually come from the human pluripotent stem cells. So this shows that we actually can inject human cells and try to analyze what will be the behavior of these cells into a real embryo. And basically, to conclude, if you go to the next slide, uh, we created this system that allows you to grow the embryos in first of all in a continuous manner, and also is very robust for up to 6 days outside of the uterus. And not only that, this culture also allows you to perform several manipulations like genetic modification and transplantation of cells, or they can also open many possibilities to study basically anything that you would like to study in mammalian development, like imaging, how the tissues grow, signaling pathways, lineage tracing. And we show that we can also study the behavior of human cells in vivo inside an embryo. And I think that the main kind of aim for us to create this platform is just to understand how organs naturally grow in an embryo, and then we can use this knowledge to build better organs on a dish. And also, just to conclude, another interesting possibility is we can also create embryos not taking them from the mouse, but kind of creating the embryos aggregating different type of stem cells that we have in culture. And these embryos are called stem cell embryo models. And now we have a platform that allows you to grow natural embryos. So maybe these embryo models will also be able to grow in this kind of ex utero system. So the combination of the stem cell embryo models with ex utero culture system will be actually something very interesting to see in the future. And basically that's it. I just want to thank my advisor Jacob Hanna and all the people who helped me in this project, and you for listening to the presentation. And if you have any questions, I'm happy to answer.
Thank you so much for that. That was fascinating, incredibly interesting. Who wants to go first with any questions? Do we have someone first?
Yeah, thank you so much, Alejandro. This is so impressive. So the total that you can go up with this rotating, so to go up to E11.5, I think you said, right? You need this
Yes.
rotating device. So uh, why does it need to be in motion? Like, do you know why, or is that just something, I don't know, was
Yeah.
done for?
No, yeah. We think that is because that first of all the diffusion of the nutrients and the oxygen when the media is in motion is always better. And for example, I don't think that the embryo needs to be in motion. I think that the medium is what needs to be in motion. So even kind of what I would like to do in the future is to create something that in which the embryo is static, but the media is flowing. I think this will also allow the embryo to survive. I don't think it's that the embryo needs the movement. I think it's the diffusion of the oxygen and the gases and the nutrients is better in motion.
So I think it would be really interesting to so or are you planning, or is the lab planning to basically expose the developing embryos to different stress factors? Let's say things related to smoking, pollutants, maybe scarcity, eliminate a few compounds for a while at different time points. I think that would be really interesting and great value for developmental disorders. Are you doing something like that or planning?
Very nice question. Yes, for our project we tested actually only one component that is called valproic acid, and this generated some defects in the closure of the neural tube. However, we never didn't test any more components, and I don't think in the lab people are planning to do it. But I hope that someone else will actually be able to use the system and test it. This is what I actually would like, that everyone or a lot of people to use the system and test the different ideas that they have.
Yeah, I think this is such a wonderful work, and will create this platform where you can, in a very controlled way, like expose developmental um during developmental stages. Like you could just like maybe apply cortisol for stress and so on. So yeah, that's very exciting. But yeah, please, Ethan, I think you have a question.
Yeah,
Go ahead.
Alejandro, thank you very much. That was a great presentation. I guess I have sort of a statement slash question in terms of, like, you were just talking about the solution moving around in terms of the nutrients and all of that. But I wonder also if another possibility would be that due to sort of the embryological development being so importantly reliant on chemical gradations at different development sensitive periods, that perhaps mixing up the solution, just keeps the gradient very constant so that the cells don't get confused and start to act further than, some envelope for which they start to migrate and differentiate.
Yeah, it's a nice suggestion, but I'm not sure if this is the case. But because what we know is that the embryos that have defects in the placenta, they also die in the same moment that our embryos die. So we think that kind of all of they need, so they are reaching a point in which, first of all, when they are small, the oxygen and the nutrients are actually able to enter into the body very easily because of the size. And then and then and this is the reason why when they are small we can keep them also in a static culture. But then as they grow, they need to be in this motion. And then after, and day 11 at day 11 they die because now they don't have, so they are too big that the oxygen is actually not able to enter into the body, and they die actually very fast, like in few hours they're already dead. And this happens also in the embryos that have defects in the placenta. So this is why we think that it's more related to the mixing and the fact that the oxygen is able to penetrate also very nicely. And actually something that we're doing now is in after E11 try to increase the oxygen below above 21%. And actually the embryos are able to survive for even longer times. So we think that it's related to that, but it's also an interesting possibility.
Yeah, what about pH balancing? Maybe it's more, a balancing of the pH than rather absolute oxygen concentration. Would there be a way to somehow stabilize the pH all over the embryo? Uh, Without using oxygen, basically, because oxygen also provides that, I think.
Yeah, very nice suggestion. So we also have add some buffers to balance the pH, but actually we never really measured it in the what is the pH level in the media itself and if it changes over time. But it's actually very interesting to check. And yeah.
Can I ask actually about your sorry, I'm trying to look at the name here. The ex utero cultural medium that you were using, yeah, the stuff, the 25% human serum that you have mixed in with there. Can you tell me what is it about that part that was useful and necessary for to be mixed with the other two parts to make this effective? And are you thinking that if you keep coming up problems with development using the serum, are you maybe going to like relook at the recipe for that, the balance of that down the line?
uh, yes, it's a very interesting point. um, so this the part of the human serum is actually so first we started with the human umbilical cord blood serum, uh, and actually this is kind of the highest quality of serum. But after we discovered that even it works with the serum that we take directly from any adult person, and we think that the reason why they need a human serum is uh, is just because uh, let's say the rat serum that we get it from a company, and maybe it's not that fresh. So there are some components in the kind of growth factors that are lost very fast during the isolation of the serum. And the fact that when we take the human serum, we isolated in the same moment that we take the blood. And this is actually already published, like from the people who invented the roller culture like 50 years ago, that when you take the serum for culture, you need to isolate it in the same moment. Otherwise, the embryos won't be able to grow. So we think that just because of the freshness of the human serum, that is what they need this component. Um, but it's also because of for us, it's also related to the price because the rat serum is extremely expensive, and the human serum basically you can have it for free from yourself. So it's also just some reason, but what we would be ideal is in the future to have a not rely on the serum, but have kind of defined components of the medium. But I guess this is for now extremely hard to achieve. So now we're just using the serum because it's the kind of a very general component and is works for now. So this is we're trying to change also a bit the medium, but until now this combination of 25% human serum is what works better.
So then, right now, it's more like other factors, and you're trying to just refine the general recipe, but it's other things rather than it won't work unless it's this component. Yeah, it's just like quality from other places and everything else you just said. Then, yeah.
Yeah, we tried many things that
Mm.
don't work. So basically, most of the things that we try never work.
Okay. And then let's do another question. I read in the paper that you gave extra glucose every 24 hours in addition to the
Yes,
medium. Is that correct?
yes,
Um,
yes.
I'm just one. I'm just wondering. Unless I'm misunderstanding, and please tell me if I am. Why were you giving? How I'm understanding it is 24 hours go by, you put some more glucose in. Another 24 hours, you put some more glucose. Why? If I'm understanding that correctly?
Yes, we increase the glucose levels, and this is because of the during development, the cells grow a lot. They proliferate like crazy, so they need also a lot of energy, and this is why we need the glucose. Actually, if you don't add the glucose, the embryos die in the day 8. So they always need the glucose, and actually now we even think that they might need even higher levels of glucose, and maybe not
And
only glucose, maybe also other components like pyruvates also help them.
Yeah,
interesting.
I know.
And
And
and that so I was just going to just finish by asking. And so that's something that you have to put in at very specific intervals. You couldn't just add like the whole amount into the medium itself or anything, and just put it all in. Does it have
Yeah.
to be given in specific bursts?
Mhm. No, actually now we already added into the medium, and we know that the even the maximum level that we add is good, is fine to add it the whole time. So currently I don't change the glucose, I just add it in the medium, and that's it. Yeah.
Ah,
But at
okay. So
that time
that's what I was
we
thinking.
didn't know that. Yeah.
Okay, thank you very much.
yeah, I was just listening to a podcast last week with a woman named Leah Giontaro, and she does like regenerative. She's trying to like get limb regeneration to happen in mammals. They've done some stuff with mice and had, but a lot of the things seem to be similar. So they um, are pretty sure that getting limb regeneration to occur is very sensitive to like the gases and the nutrients like you were talking about. Did that come from you? She also said that they haven't really been focusing on the amount of gases, but that what they do know so far is that excess amounts of nutrients is super important, as well as like so the glucose, but also they said having low oxygen levels at certain times is very important, as well as having insulin, and that they can actually get some of these different model organisms to start regenerating just from having a lot of excess nutrients in the glucose and insulin, and then sometimes playing also with the levels of oxygen. Does that come from, do you know, does that come from this kind of stuff in embryogenesis, or did this discovered independently?
Mm, I'm not sure. I will say that it's they discovered it independently. I don't know actually that much about regeneration, and in mammals, it's really not kind of a field that I have seen a lot. Um, no, I cannot. I think I cannot comment much about that more than this. Yeah. But indeed, like it's very important. Like all this metabolism. is I will say that for me is the most important uh, factor of this trying to find this system to grow the ex utero. I think the metabolic aspect is the most important.
Yeah, Dr. Shagwe.
Thank you, Carolina. So thank you, Alejandro, for sharing your fascinating paper with us. My question is about the inequality of the mitochondria because I'm not sure. Did you guys just take out all of the mitochondria? I mean, before just transfer that or not? All of the cells still includes mitochondria.
No, all the cells are normal. We don't modify anything of the embryo, so they all have mitochondria. Everything is normal. They're wild type embryos.
And
Yeah, we never modify.
sure, go ahead. Okay, you want to explain more? I just waited that you can complete your sentence.
Oh, yeah, go ahead.
Mm, so just I'm trying to kind of understand the question. So you want to know if we remove the mitochondria from the cells?
Yes.
Uh, yeah, no, but as I said, no, 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 we don't remove the mitochondria. I think this if we do that in some knockout of some mitochondrial gene, the embryos will die immediately. I don't think they are able to survive that because
Okay.
they require a lot on the also on the TCA cycle. We know that in the late organogenesis, like after day 10, they require also a lot of this. So I don't think they will be able to survive.
I see. So I was just wondering because you just transfer from the vitro to the another part, and I was just wondering maybe beside the experiment you had this experiment to extract the mitochondria, or I mean, for get a better information around the quality of the mitochondria or such a thing. And also in a slide that you just explained about two clusters, which I mean about the consistent with insufficient oxygenation and nutrient supply, because we see the erythroid lineage and myocyte. Would you please explain a little bit more about why you mentioned insufficient oxygenated and nutrient supply? You want to put a stress, or what was the main reason?
Uh, that's a nice question. So the reason is so it's known from also from previous studies on culture of mouse embryos that the main reason why the embryos die uh, in the late when they become very big is because they cannot depend anymore on the use diffusion to for the oxygen and the nutrients to enter into the body, and this actually so we know that some of the main organs that are first affected by this kind of insufficiency of oxygen are the blood cells and also the heart. And actually, there was a very recent paper published like 1 week ago in which they analyzed the metabolism of the mouse embryo, and they do a knockout for the some component of the TCA cycle, and basically what they see is the same that we see in our system, that the embryos have an affectation in the heart and the uh, red blood cells. They also see it in that in this knockout. So we think that is basically this is related to the fact that the embryos are already suffering because they are too big, that the cells don't get enough oxygen and enough nutrients. So this is why they die and why we see the changes in the gene expression in the red blood cells and in the heart. It's basically metabolic reason, and it's known that also the embryos with problems in the placenta, they will die at this is also kind of the day 11, and because of the same reasons, because of the insufficiency in oxygen and nutrient transportation, and they will also have first problems in the heart and in the blood.
And so the TCA cycle that you just mentioned, so did you find any further information from TCA cycle, for example, any inhibition, or the first part was more active or second part, and how was the ratio and production of the ATP out of the TCA? Do you have any more information to release with us?
This I don't have more information, but I can actually share with you the paper that I that was published last week, so maybe you can find something there.
Sure, that would be wonderful. Thank you.
Do we have any more questions? Has anyone else got something to ask? Let's have a look. Okay, yeah, let's do that.
Thank you.
Mhm.
Yeah, if nobody else has, I have just more comment. It was really impressive how the throughput was for how you could get so many cells with the genes of that you wanted to insert. I think that was really impressive, and this will be another great platform to use to induce or delete specific genes in a very efficient way, and then see how the developmental output was. So yeah, it's really impressive work, and I assume it was a lot of work. So how long did it take to get this paper out? Like, yeah, how many years of work was this for you?
Thank you very much for your kind words. And so I've been in the lab for 5 years and working this a lot of time. Where actually another person was also working even before me for like 2 years. So it's like basically there for, let's say, about 6 years more or less maybe. But yeah, like it's takes time, but this is not easy to solve a question.
Yeah, I
You
just
still feel
wanted to mention it because I think people see one paper of a few pages, people that are not in research, just to give them how much work, how many years of work, those pretty figures and that paper is actually like how much is behind it. And before that, your PI and maybe you included also were writing the grant for this for a few times. So it's a lot of years to get to a nice, wonderful paper like this. So yeah, thank you.
And are you feeling that your progress with this is a slow and steady progress, or do you have times where you hit a wall and you're kind of stuck for a short time without a breakthrough? How does it go?
So I think that most of the times actually we don't advance. Uh, so most of the time, as I said, most of the times that we try, they never work. It's rare that something that we try works. Um, so yeah, just I think just well, I don't give up, and it's not that easy. I'm trying to stay in the culture as much as I want, but it's really hard to find new ideas of what to change.
You must have to be incredibly patient and persistent then, especially if you're trying loads of things and it keeps hitting the wall and you're trying to come up with new permutations, yeah?
Yeah, I have to be tolerant to frustration, I think, because most of the time the experiments don't work.
Tolerant to frustration. I like that's actually a really, really good term for that, right? Because of course, the next breakthrough that you make with that just brings with it a whole new set of things to discover, right? And exploring new possibilities.
Yeah, indeed.
Are you familiar with the movie Room with a View?
No, no. What is it about?
I am not familiar with it. You can explain about it if you want.
Well, this is absolutely fascinating. Like, where do you see this going? Is the goal then to try and get this to a full mouse developed after the 19 days? This is like your probably sort of longish term goal, right? To get the mouse out of the womb, sorry, artificially created, and then are you going to try with other mammals, or which direction do you see the whole thing going to eventually?
It's a very interesting point. Well, for me, the most important thing of the culture is to kind of to be able to understand development. Of course, I would like to see the mouse kind of being born from this kind of system, but I think that is still too far away. There are too many things that we need to uh, kind of to solve first. Mm, and I don't know, maybe I would like at some point to do it with baby monkey, but I think we will not see these kind of things in the next 20 years, because I
Hmm.
think it's very hard.
It sounds very hard. You also mentioned that the process you're describing, that so far the development's not been successful enough, but at the later stages it requires like a uterus substitute much more. Is that right? Like in the last few days?
Mm, so what I mentioned is that in the early stages, like from day 0 to 4, because the embryo is not implanted in the mother, so you can actually take them out, and they are not that dependent on the mother. So people have done a lot of experiments with the pre-implantation embryos, and then you can just put them back into another mother, and they will grow. So that's the reason why we know a lot more about pre-implantation than about post-implantation, because after the implantation, then the embryo becomes dependent on the mother. So basically, if you take them out, they will die in one one day maximum before. But now we managed to extend it for some time, for up to 6 days. So I hope that now it will be easier to understand more development. But yeah, we'll see what happens in the future.
So does that mean then that everything you're doing at the moment is to get it to that point, and then it'll be a new set of challenges when you have to give it like an artificial uterus and everything? That'll be like a new stage of challenge there. Yeah.
Yeah. Because after we need some kind of artificial placenta, and then this is another challenge. It's completely different. I don't think the system that we have will manage to grow an embryo until the end. We need to change it.
Oh, that's interesting. So you're already doing it knowing that this all going to be changed along the way. If that's the case, then it's sounding like there'll be a, a very interesting plus and minus here. And the plus being, if you can actually have it connect to an artificial uterus, you will be able to actually observe the development at that point, right? You know, the part that you said up until now, we can't watch it because it's in the mother and it's connected and everything. But if it's artificial, then we will be able to observe that. But of course, the downside to that is, since we don't know what's going on in there exactly, we don't know how to properly make a uterus that could do that. Is that right?
Yeah, yes, indeed. At some point, for example, there is one already paper in which some people managed to grow a kind of late fetus of a lamb inside some kind of bag and some artificial uh, circulation, and maybe we had at some point the idea kind to combine that system with ours, but this is still too far and still extremely hard because the mouse embryo is very small. In the case of of this sheep that they did is because they already had a very big fetus, so it was easier to connect to this artificial circulation. But in the case of the early embryos, it becomes a lot harder, and maybe also not the same system will work. So it's a lot of complications there.
That must be difficult then when you're seeing potential in other papers and you're thinking maybe there's a linking part. But of course, by the time you're at that other stage, they will be at another stage too, along with perhaps others that you'll be able to cross over with, right?
Yeah, yeah, indeed. Um,
Uh,
I don't know.
jo
Yes,
I'm sorry that I interrupt. Josie, she had the question. She came up to the stage. I wanted to give her a chance before closing the room.
I came up more to just commend you on your work. I think this is a really important area of research, understanding the development of an embryo more closely and making it so that we can look at it more closely. Yeah, it's just amazing. Thank you.
thank you very much.
Yeah, this is absolutely incredible. I'm actually quite excited about where you're going to be able to go with this, and I'm looking forward to seeing how you're going to solve these problems, eh? Because it's always great, exciting when you get a breakthrough. So please, for the sake of all of us, please keep persistent. Please keep resistant to frustration.
Thank you very much for your words.
Yeah, thank you so much. I totally agree. This will give us a lot of insight into developmental biology, into different developmental diseases, and it will be an amazing platform for that. And I really appreciate you taking the time to answer our questions. Yeah, thank you so much, Alejandro.
No, thank you for the
Thank you
invitation.
very, very much.
Thank you, and see you soon. Then I hope to see you at some point.
Yeah, come back
Absolutely. I hope
anytime and with
anytime you get a
updates.
breakthrough,
Yeah, or just come and join our discussions when you have time at around this time. We have a few rooms that we have in our, like in my early morning lunchtime. This is for me lunchtime. So yeah, I hope you find interest in what we do here. And yeah, and please come back with any updates, or just we can just have, I think, another room about this. We can keep talking. So yeah, and everyone, thank you for asking questions. Join the Science Society, and we will have more rooms like this with guest invited speakers. Then, yeah, and I'm sorry I'm in the car in New York City right now because I had to leave. So sorry for the background noise. And yeah, looking forward to hearing you. And Alejandro, thank you again. This is such an amazing work. Congratulations.
Thank you very, very
Thank you very
much.
much. Bye.
Okay, bye everyone. 3, 2, 1.
Bye,
Bye.
everyone.